So I'm excited to have Reed Johnson with us this morning, a faculty member here in the Department of Entomology, doing a lot of awesome research related to toxicology, honeybees, waggle dances he was talking about this morning. Some great research here on the Worcester campus as well as across the state. And so this morning, Reed is going to talk about viruses in honeybees and how we can help protect our bees. So Reed, I'm going to turn the floor over to you.
So, I was assigned the topic of viruses to talk about, and I'll start. I should never start off with an apology, but I've worked some with viruses, but this is not really my main area of research. So I've kind of gone and reviewed the literature, and that's what I'm going to present to you today. If you have questions, it's reasonably likely I won't be able to answer them, because viruses are somewhat less mysterious to me than they are to you. I think it's something that certainly I know I need to learn more about, and I know all beekeepers should really appreciate the importance of viruses in bee health. So, bees are dying. We certainly experience this here in the OSU apiaries. These are the average winter losses per beekeeper from the Bee Informed Partnership over the years from 2008 to 2016. And you can see that it's grim. It's hard to keep bees alive. We very commonly lose half of our colonies in our apiaries for unknown reasons. Certainly we could probably do better with our Varroa control. We could probably maybe feed them better. And we're doing research on them, which is not necessarily good for bee health. But these kinds of numbers are quite unsurprising for me. This is certainly maybe even a little bit better than what we experience. That it's not uncommon to lose half your colonies over the course of a year. And it's depressing. I don't like it. I don't like my bees dying, and I really would like to know what the cause behind this is. Here in Ohio, you see we have, this is from the last year, we have a 39% annual loss in Ohio, as you can tell by the dark orange color. That's not as bad as some other states that are in deep red, but still not great. So the Bee Informed Partnership, if you haven't ever gone to their website, I would really encourage you to go there and to participate in their surveys. It's a phenomenal resource. It's great to know what beekeepers are doing and what kinds of success they're having with the different techniques that they're using. So one of the questions in the Bee Informed Partnership's survey is, why did your bees die? And beekeepers come up with all the usual answers. Queen failure, starvation, Varroa are at the top of the list, as well as common other problems: nosema, small hive beetle, poor wintering, pesticides, which is the part I'm particularly interested in, weak colonies, whatever that means. It's only down here at the very bottom that you actually get to beekeepers saying that their bees are dying because of viruses and other diseases. Viruses don't even get their own category on this chart. And even if they did, it would just be a minuscule number of beekeepers that are reporting that viruses are the cause of bee mortality, of the bee mortality that they're seeing. And I am almost 100% sure that this is not the case, that viruses are actually responsible for a lot more of the mortality that we're experiencing. It's just that we don't really appreciate the role that viruses are playing in our pretty dismal ability to keep our bees alive from year to year. So I'm going to outline some virus basics and the taxonomy, the symptoms that these viruses produce in bees, the most important thing, which is this interaction with Varroa, the Varroa virus interaction, and how we can control viruses, possibly by controlling Varroa, even though that may not be 100% effective. And then finally, at the end, I'll go into how, if you wanted to detect viruses, so you could truly appreciate what role they're playing, how you would do that. Unfortunately, it requires a molecular biology lab, but I'll show it to you anyway because it's good to see what needs to be done to really detect viruses in bees. So the main problem with the viruses is that they're so stinking small. They are tiny, tiny little, things, and it's not even clear if they're alive. But as you can see, here's a size scale. Up animal cells are, tens of micrometers. Bacteria, 1 micrometer. Viruses are in the submicrometer range, 100 nanometers. They're so small that you cannot see them with a light microscope. So what you use for nosema detection is never going to work for virus detection. You need access to an electron microscope or X-ray crystallography, as was finally done with deformed wing virus here in 2017, or the molecular approaches that I'm going to show you at the end of the talk. Viruses are a difficult thing to study. You know, this deformed wing virus, as I'm going to tell you in a minute, is the most severe virus, the worst virus in honeybees, and we only finally got an image of deformed wing virus this year. In 2017, it was finally published. So viruses are difficult to see, they're difficult to work with, they're difficult to understand. And that's this big cloud that really separates us from really understanding what the viruses are doing in bees. So just to understand viruses, we have to go back to some basic biology, and I hope not to put anyone to sleep at this early hour in the morning. Hopefully you have a nice large cup of coffee next to you so you can keep up with the biology here. So here's the central dogma of biology. And that central dogma is you have DNA, and DNA, of course, is the genome for all animals. Humans, bees, everybody has DNA in all of our cells, and that DNA is transcribed by an RNA polymerase into RNA. So DNA is kind of the backup copy of your genetic material. RNA is the working copy. That's what actually goes through the ribosome to produce the proteins that make up your cells, that make up all of the other parts of you. So it's this central dogma says that we go from DNA to RNA to protein, and that's the way life works, and that's the way we get our cells and every organism survives on this planet is through this central dogma. Except for viruses. Viruses are different. Viruses have a genome that could be RNA. So most of the honeybee viruses are in this group of positive sense single-strand RNA viruses. And they actually store their genetic information as RNA. They don't have DNA inside the viruses that your honeybees get. It's all RNA-based viruses. And this may seem like a trivial detail, but this is actually vitally important to understand the difference between how our cells function and how these viruses function if a method of control is going to be finally deployed and understood. So viruses are a very different form of life, in quotes, if you can even call them that, because they use RNA as their genome, as opposed to all other life. So here's a family tree of the positive sense single-strand RNA viruses. And this is from that same paper that finally gave us a structure for the deformed wing virus. And this is just a family tree here. Here we have deformed wing virus right here. Deformed wing virus is an iflavirus, as you can see in this yellow colored pie wedge. And it's interesting to see what deformed wing virus is related to. You know, up here in the apivirus group, we have the Israeli acute paralysis virus, another potentially serious honeybee virus. There's other insect viruses. Here we have a cricket paralysis virus that's kind of in between deformed wing virus and Israeli acute paralysis virus. But if you look at the tree here, there's also viruses in a lot of other organisms. Here's hepatitis A virus, which infects mammals and humans. Human parechovirus. Foot and mouth disease is down here. So single-strand, positive sense single-strand RNA viruses are the most common honeybee viruses, but this group of viruses also infects, most other living things and can cause really serious problems in humans and are a very serious problem in human health as well as bee health. And as a result, there's actually quite a bit of research that goes into this group of viruses, because obviously hepatitis A and C, which are positive sense single-strand viruses, are really serious problems, and there's a lot of medical research that goes into trying to figure out how to control them. So let's look at how viruses reproduce in the cell. And there's some great YouTube videos on viral replication. I'm going to take hepatitis C virus because it's one of these positive sense single-strand viruses, just like the deformed wing virus and other viruses that bees get. Let's look to see what one of these viruses is doing inside the cell. You can go watch this YouTube video after the webinar today, and you can see this in all its its wonderful detail. Unfortunately, I can't rebroadcast the YouTube video here, so I just took some screenshots to show you what's going on when one of these viral particles, which are very tiny, invade a cell and how they get in there and reproduce. So here we have a viral particle. This is floating around in the human bloodstream, or it could be in the hemolymph of a bee or in the gut. Looks pretty innocuous. Inside of it is its RNA genome, wrapped inside this layer of proteins and a lipid bilayer. So that viral particle is floating along, and it encounters a cell. And you can see the massive size difference here. Here's a red blood cell, because this is a hepatitis C. Here's this little tiny virus particle, and here's some of the other cells in the human body. And it attaches to the surface of that cell and kind of injects itself inside the cell using a number of proteins on the surface of that host cell, basically hijacking the cell to let it in. And then once it's inside, it will release this RNA genome from the viral particle. That RNA genome, because it's already RNA, it can be directly translated into proteins by hijacking the cell's ribosome here. The ribosome, of course, is the cellular machinery to take that RNA, use the code in that RNA to build the proteins that would normally be for the cell's use. But because this is a virus, it's hijacking this ribosome, and it's going to have this ribosome make proteins for more viruses. And there's a number of different proteins encoded in this RNA genome here that it needs to build in order to make more viral particles, which of course is the whole point here. So once it makes more viral proteins, one of those proteins is an RNA polymerase, which forms this RNA replication complex, which then takes the virus's RNA genome and replicates it. So you see on the left here in green, we have this positive sense single-stranded RNA genome from this virus. It goes through this replication complex and gets turned into double-stranded RNA with a positive strand and a negative strand. At this point, it looks a lot like DNA. It is very, very similar to DNA. And this is just making kind of like a photocopier. You're making multiple copies of this RNA genome, which can then get packed inside these new viral particles. Here we've got all these viral proteins that were created by hijacking that ribosome, plus a new fresh copy of the virus's RNA genome getting encapsulated here in a new viral package. And then you get this package built with the RNA genome inside. And then eventually that will get released back out. The cell will make thousands of these copies of these RNA particle or RNA genome, the viral particles, and then these will eventually get released and can go infect other cells and eventually probably cause great harm to the liver in the case of hepatitis C, or kill your bees in the case of something like deformed wing virus. But the basic biology of reproduction of these viruses inside the cell is essentially the same for this entire family of positive sense single-strand viruses. We'll come back to some of this biology because that's where we can start talking about how to control viruses. So I mentioned that there are a number of different honeybee viruses, and almost all of them are these positive sense single-strand viruses. There are at least 24 viruses that have been named. It's probably more than that now as people continue to discover more and more of them. It's kind of difficult to categorize viruses because they are evolving quite rapidly and they're changing, and it's not really easy to, the taxonomy of them is not easy to understand because many of them are very closely related. There are 16 to 18 distinct virus groups within this broader group of honeybee viruses. Many of these viruses were actually discovered using electron microscopy back in the 1960s through 1980s in the UK. The classic work done by Bailey and Ball that really showed us that bees had viruses at all. At that time, bee viruses were kind of more of a curiosity. You could find bee viruses, but they did not seem to be killing bees. Occasionally, you'd find bees or maybe a colony that would die from an overt viral infection. But viruses didn't really seem to be much of a problem back then. Kind of more of a interesting phenomenon, biological phenomenon to be studied. But that's where we historically have understood most about viruses. Today, we don't use microscopy to discover viruses anymore. We use DNA sequencing, or RNA sequencing in this case, to create DNA, which is then sequenced to identify all these new viruses. And there's undoubtedly more left to be discovered out there. So, I mean, it's very clear that if a bee has a high level of virus in it, that's not good, and bees can die from viruses. This has been known since the 1960s from the classical Bailey and Ball work. This is work that was done back in 1999, and this is just the optical density, which is a way to measure the number of viral particles in a bee. And you see if you have uninfested, non-deformed bees, they have relatively low number of viruses. If you have viruses or bees that are infested with Varroa, you get maybe a little bit higher levels of virus. Mites also have these viruses in them. And then when you start to see sick and dying and dead bees, often these dying and dead bees or mites have high levels of these viruses in them. Now we don't know which virus this is because this is just based on optical density. But this is really foundational work showing that viruses are more abundant in dead and dying bees and mites than they are in otherwise uninfested healthy bees. Now, one of the bee viruses that is probably the one that we should really be paying the most attention to is Deformed Wing Virus, or DWV. This is the one that I showed you that we finally got a picture of it just this year. And this is called Deformed Wing Virus because it produces this classic twisted wings symptom when a bee has an overt infection. Deformed wing virus is, a complex of viruses, really, and there's a whole number of related viruses that share great similarity with deformed wing virus. There's the Kikugo virus, which has been associated with elevated aggression. There's the Varroa destructor virus, which is specific to Varroa, but is closely related to deformed wing virus. And then there's slow bee paralysis virus, which is related to trembling hairless adults. It's a whole suite of viruses that seem to be closely related to deformed wing virus that can cause various kinds of symptoms depending on the particular virus. So here's that classic example of an overt infection of deformed wing virus in bees. And this is a pretty rare occurrence prior to the introduction of the Varroa mite. So now, if you see one of these young deformed wing virus bees emerging from a cell in your colony, that deformed wing virus bee probably has 24 to 48 hours to live at the most. This kind of bee is not going to ever do very much for the colony. It's essentially a walking dead. It's essentially a big bag of viruses that will potentially go on, infect many more mites and many more bees, and spread that viral infection. These other bees in this group, the chronic bee paralysis virus, this is the one that produces this trembling bees and hairless black syndrome, which can sometimes be confused with pesticide kill. Chronic bee paralysis virus also can infect larval and pupal stages to varying degrees, but it does not appear to be as severe as the deformed wing virus. So it's pretty clear that as we have been able to discern these different kinds of viruses using the methods that I'll show you at the end of the talk, dead and dying colonies have more virus than healthy colonies. So this is work that was done back in the late 2000s, showing that dead and dying colonies have higher levels of deformed wing virus in gray here, as well as acute bee paralysis virus. And there's a pretty clear correlation between the number of viral copies in these dead and dying colonies and that colony's status, and that healthy colonies generally have less deformed wing virus, less acute bee paralysis virus than healthy counterparts. This is more evidence that deformed wing virus is really associated with colony failure. So deformed wing virus you can measure it in that kind of way, or you can measure it with this kind of visual overt symptoms. Obviously, deformed wing virus can kill in the adult stage because these adult bees with deformed wing virus are not going to live more than 24 to 48 hours. But it's actually interesting to know that deformed wing virus is essentially everywhere in all bees, but not all bees display this kind of overt infection. It's present in pretty much all castes, all life stages. It tends to be more abundant in the head and the abdomen, but the only obvious sign that there's a serious infection is when you see these twisted wings. But that's not to say that this virus, in the absence of seeing these overt infections, is entirely harmless. There's newer work being done to show that these covert infections that don't result in twisted wings actually have negative consequences for the colony as well. So this is a nice study that was published just this year, and they wanted to see what kind of effect does this covert infection, not resulting in twisted wings, have on bees. And so what they did is they put these bees in an observation hive, and they injected bees with either deformed wing virus or a control, which was the deformed wing virus RNA, but without the viral coat, so that these viruses weren't really able to reproduce in the bees. But that's a good control. And then they attached RFID tags to these bees so that they could monitor the amount of foraging that these bees are doing. And the way these RFID tags work is that in front of this observation hive, they have an RFID tag reader, and this is very similar to the newer kinds of, you've been at a hotel recently, the newer hotels have the kind where you just wave your key card in front of the door lock, and it opens the door for you kind of magically. Well, RFID tags are the magic inside that those newer hotel key cards, and you can put the same kind of chip on a bee so that when the bee leaves or returns to the colony, there's this RFID antenna reader here that is picking up that particular bee's departure or re-entry to the colony. And you can use this approach to look to see, how often bees are foraging, and critically for this experiment, what age those bees begin foraging at. And so you can produce some really interesting results with this. And what this group found is that if you inject bees with deformed wing virus, those bees begin foraging earlier. So here we have bees in 3 colonies with deformed wing virus in gray here, and without deformed wing virus in white below. And we've got the day at which those bees were foraging. Now, they affixed these RFID tags to these bees when they were very young, so they did not begin foraging for several days because obviously very young bees take on the nursing duties in the colony, and they don't leave. They're taking care of the larvae. But what they found is that bees with this deformed wing virus injection began foraging earlier than their counterparts that received the control injection. And they saw this consistently across all 3 colonies, that the deformed wing virus infected bees with this covert infection, though they didn't have the twisted wing syndrome, they had more virus in them, and they were beginning to forage earlier. Now, the consequence for a honeybee of beginning foraging earlier is that means they will also die earlier, because foraging in honeybees is a death sentence. And once they begin foraging, they have, on average, about a week left to live. And so it's no surprise that bees that were injected with deformed wing virus experienced higher early life mortality and decreased lifespan because they graduated to foraging sooner, and that means that they die sooner. And that's going to be true whether you have deformed wing virus or not. If bees are foraging prematurely, they're just not going to live as long. But this is one consequence of a deformed wing virus infection that may not be visible, but is still having negative consequences on your colony, even though maybe you don't actually see any bees with twisted wings. Just the presence of that virus can be having subtle and harmful effects on the health of that colony. Now, I mentioned Varroa a minute ago, and this Varroa deformed wing virus association is probably what's driving most of the problems that we're seeing with Varroa and with bee health in general today. So this was really a groundbreaking study done by Stephen Martin, who's a researcher in the UK. It was published back in 2012 in Science, and they took advantage of the fact that for many, many years, the islands of Hawaii were Varroa-free. Honeybees were introduced to Hawaii in 1857, and they were kept there very successfully for over 100 years. It was only in 2007 that the Varroa mite was somehow introduced to the island of Oahu. And that's been obviously very devastating for the queen rearing industry that is out there in Hawaii. And then in 2009, it had made it over to the Big Island and was beginning to take over there. And I believe it spread to additional islands in Hawaii as well since this study was published. But this was a really excellent laboratory in which to study this interaction between the Varroa mite and deformed wing virus. Because interesting thing is that deformed wing virus was present on the islands of Hawaii long before Varroa was ever introduced. But that presence of Varroa kind of enabled deformed wing virus to become a problem. And this chart here really tells the whole story. And I'd encourage you to go take a look at the science paper if you're really interested in it. So here on the bottom, we've got the different islands of Hawaii. Oahu is on the right here, which at the time had Varroa for greater than 3 years. There were different populations on the Big Island of Hawaii, which had experienced Varroa for between 2 and 0 years. And then they looked at colonies on other islands that had not yet had Varroa introduction. And what they found is that bees on these islands with Varroa had much higher levels of deformed wing virus in bees that they looked at. More copies of Varroa or of deformed wing virus in bees on islands where Varroa was present. And down here on these pie charts with the red on them, you can see that the prevalence of deformed wing virus really just took off like a rocket once Varroa was present. It's present but relatively rare on islands without Varroa, but once Varroa was present, it becomes essentially ubiquitous. All colonies had deformed wing virus once Varroa was present. So Varroa is increasing the prevalence of deformed wing virus, but it's doing more than that. It's changing it's changing deformed wing virus itself. So prior to the introduction of Varroa on these islands, you can see that deformed wing virus strain diversity, deformed wing virus, like other viruses, comes in a lot of different quasi-species. Like we talk about H1N1 flu is one particular quasi-species of the flu virus. Deformed wing virus also has a number of different strains that are naturally present. But the presence of Varroa mites selects for one particular highly virulent strain of deformed wing virus. And as you can see, the longer that Varroa is present on an island, you really lose diversity of these deformed wing virus strains. So you're just left with this one highly virulent strain. And it's the same strain that is probably highly abundant in our bees here on the mainland United States. So I mentioned deformed wing virus has different strains. It could be, and there's a lot of work going into this now, which strains are worse, how is the Varroa mite associated with these particular deformed wing virus strains. One strain in particular, this deformed wing virus B, is strongly associated with Varroa and overwintering mortality. And this is a study that was again just published this year, looking at the prevalence of deformed wing virus strain B. This is one of the highly virulent strains, and you can see that as down here on the x-axis we have colony mortality, and we've got pathogen prevalence on the y-axis here, and you can see that as there's a correlation here between the abundance of this deformed wing virus B strain and the likelihood that that colony is going to die. Interestingly, this was not the case with black queen cell virus, another very common honeybee virus. So this just drive home the point that there are particular strains within deformed wing virus itself that are probably tightly associated with Varroa mites, and they are kind of have this complementary relationship with Varroa mites, so that the Varroa mite is vectoring these viruses, and these viruses are enabled by the presence of that Varroa mite, and it produces a really bad scenario. So to get away from deformed wing virus and relatives for a minute, we'll talk about another group of honeybee viruses. This is the acute Kashmir-Israeli complex, or AKI complex. This includes the acute bee paralysis virus, Kashmir bee virus, Israeli acute paralysis virus, and the black queen cell virus. If you'll recall, Israeli acute paralysis virus was the one that was originally associated with the colony collapse disorder phenomenon back in 2007, 2008. I mean, it can still be a deadly virus, but it does not appear to have the horrible consequences that deformed wing virus has. This complex of bees, or of viruses rather, will kill bees. There's no question about that. But it has the advantage of being more deadly. And that sounds counterintuitive, that you would want a more deadly virus. But you do. You want a virus to actually take out your bees so that you don't have these bees surviving the viral infection to go on and allow that virus to reproduce and to spread to other colonies. It's almost better if it just kills them, and then that virus will die with it. And these viruses have been associated with Varroa as well, but these viruses are probably bad for the mites in addition to being bad for the bees. So these are bad viruses, but they do not appear to be as bad as the deformed wing virus types that are associated with Varroa mite infestation. So here's just some examples of viruses you may encounter. I've just seen this a couple times in my life. Black queen cell virus. This is one that attacks larvae in particular, and you may see queens turning black. This is a classic symptom of a black queen cell virus infection. You can get these symptoms in larvae. These will be present. This virus will be present in adult bees, but there'll be no obvious symptoms with a low-level infection. Now, these two groups together, it certainly could be that deformed wing virus and these particularly virulent strains kind of opens the door to additional viral infection, because there's certainly a lot of viruses out there. And it could be that after deformed wing virus gains a foothold, other viruses move in. And that was certainly the result of the great amount of work that went into understanding the CCD phenomenon back almost 10 years ago now. And in every study that was done on CCD, they always found viruses in these collapsing colonies, whether it was Israeli acute paralysis virus, the Kashmir bee virus. They just, in my own study that I did as part of my PhD dissertation, we found increased levels of deformed wing virus and just more viruses in general in colonies from the classic colony collapse disorder group. So it's really a consistent finding that dead and dying bees have more viruses. But it's increasingly clear that it's deformed wing virus that is likely opening the door to these other viral infections, and that's the one that is likely causing a lot of these problems. Just some other viruses. There's the sac brood virus, which you may see as this kind of gooey sac. Again, this is one that has symptoms that are present in larvae, though it can be detected in adults as well. Now, I should mention these viruses with covert infections in adult bees could well be having consequences in hive performance that we just don't know about. We really don't know that much about viruses and their effects on colonies aside from death. And it's entirely possible that these viral infections are having negative consequences at a sublethal level that we really don't appreciate yet. But from everything we see, it's deformed wing virus and that Varroa association that's the most clear killer. Now, viruses can be transmitted in all manner of ways between different bees. There is a sexual method of transfer. They can be transferred in the semen when drones mate with queens. They can be transferred vertically when a queen who's infected with a virus lays eggs, and then those eggs will be pre-infected with a virus as they are developing. Then, of course, there is possible for adults to transmit viruses as they are caring for the larvae and feeding them, or to pick up a virus, potentially as they're cleaning out dead bees that died from a viral infection. And, these routes of transmission have been going on forever, and this huge diversity of viruses probably use all of these different routes of transmission. Vertical, horizontal, sexual, many different routes for a virus to get around in a bee colony. But it's this introduction of Varroa which opened up a new method for viruses to be transmitted between adults, which is critical, and between pupae with the Varroa mite as a vector, between adults and pupae, and between colonies when those mites will drift. Interestingly, viruses are not limited to a honeybee colony, and many of these viruses that we associate with honeybees are actually present in the environment. And this is kind of a interesting study done by Singh et al. He was at Penn State at the time, and they looked at the viral infection rate of foragers from a honeybee colony, and they could find black queen cell virus, Sakura virus, and deformed wing virus in a number of the foragers that they looked at. But if they looked at the pollen that these bees were collecting and bringing back, you could also detect all of these viruses in the pollen bees were handling as well. And this raises the question, are these sick foragers transmitting their viral load to the pollen as they're collecting it? And then this infected pollen will go on to feed nurse bees, and those nurse bees could potentially become infected with the viruses that the foragers had just through consumption of pollen, not through any other route. It also raises the idea that these viruses are not limited to just honeybees. You can certainly find what we would consider honeybee viruses in a number of other insects that are visiting the same flowers that honeybees are visiting. Common eastern bumblebee, paper wasps, and yellow jackets that are visiting flowers for nectar also have deformed wing virus, Israeli acute paralysis virus, and black queen cell virus detected in them. And whether this is a major concern or not is really an open question, because it's entirely possible for these honeybee viruses to be transmitted to all of these other wild insects and potentially cause them harm, or the route could be the opposite direction. It could be that maybe bumblebees are a major repository for deformed wing virus, and it doesn't cause real negative consequences in a bumblebee, but if a honeybee picks up that virus from a flower, that could potentially greatly harm the honeybee colony. So these viruses can clearly reproduce in other insects. They're not limited to honeybees, and the consequences of this interspecies transmission are really unclear. It could be harmful for honeybees, it could be harmful for all these other wild insects, or it could be just harmful for everybody. In addition, viruses are crazy things. There's a plant virus, the tobacco ringspot virus, which is a problem in tobacco agriculture, which has been found in honeybees. And this work done by the USDA lab in Beltsville a few years ago found that colonies were more likely to collapse with this suite of tobacco ringspot virus. This is this virus that was only known from plants previously, in combination with deformed wing viruses as well as other viruses, caused increased colony collapse. So maybe it's not just insects, that plants are involved in this whole viral thing as well. But I guess getting back to the question of how do we control viruses, and the obvious answer is through controlling the mites that are this new vector for viruses. Previous to the introduction of Varroa mites, viruses were kind of an academic thing, that people didn't have too many problems with viruses, and relatively few colonies died from virus infestation. And it's definitely true that Varroa destructor, if you can control your Varroa numbers, you can decrease colony mortality. This is from one of these papers that I already showed you. You can see that if you have lower Varroa numbers, you have lower honeybee mortality in colonies, and these acaricides, the miticides that are used for killing Varroa mites, reduce result in decreased Varroa mite infestations, therefore resulting in increased bee survival. And this holds up in national surveys. Again, going back to the Be Informed project, where they surveyed beekeepers across the country for many years. You can see that in the United States, beekeepers who used a Varroa treatment only suffered 32.5% average winter loss, versus beekeepers who did not use a Varroa treatment suffered 42.3% loss. So that's a 10% reduction in the overall reduction in the number of colonies lost if a Varroa treatment is used. So there's no question that controlling Varroa mites leads to better survival, but the question is, in controlling Varroa mites, are you really controlling the viruses that appear to underlie a lot of the problems that Varroa mites are associated with? And that answer is not clear at all. Studies have been done looking specifically at the virus level in colonies that have undergone mite control. So these are colonies that were treated with the miticide apistan, which was effective in this experiment at controlling Varroa mites. It may not be effective for controlling mites in all situations, but what they found is really kind of a disappointing reduction in the level of bee viruses. You have deformed wing virus here, and in the treated colonies, which is the the dashed line, you do see a reduction in the amount of deformed wing virus in these treated colonies, but not as rapid or as great as a decrease in deformed wing virus as you might hope for. And black queen cell virus was entirely unaffected by control of Varroa mites. So it seems that Varroa mite control is the best we can do for controlling Varroa, or controlling the viruses that are at the root cause of a lot of bee problems, but it's not the whole solution. Varroa control is kind of an incomplete control for viruses, because the viruses will exist even if the mites are well controlled. And even more worrying, this is a paper that was just published out of Stephen Martin's group in the UK, and they studied a survivor population of bees in Brazil that had never shown any problems with Varroa. They had Varroa were present, but those bees appeared to be tolerant of the mites that were on them. Varroa in this group of bees wasn't killing the bees. And the question was, what's the mechanism behind this tolerance of Varroa mites in this population of Brazilian bees? And what they found is that the key thing here in this population of bees is that they did not have this virulent strain of deformed wing virus. That virulent strain was present, but an overt infection in this population of bees apparently had never occurred, and that an overt infection is really the lighting of the fuse, and that once you have an overt infection, that overt infection with the twisted wings, that virus will then get into the mites, and that begins this cycle of mite transmission and the downward slope and eventually colony failure. So their conclusion was that this Varroa-tolerant population of bees in Brazil is really on borrowed time, and it's just a matter of an overt infection of the deformed wing virus strain that's already present in the population to interact in the right way with the Varroa, which are already present in this population, to free that Varroa virus interaction to take out this population of bees, which is really kind of a depressing finding, that it's nothing special about these bees or about these mites in particular. It's just the fact that they have been lucky. So, direct virus control. If mites are not the entire answer here, how can we get at the virus problem directly? Well, there are drugs being developed for this group of positive single-strand viruses, such as the drug Sovaldi, which is very effective at controlling hepatitis C viral replication in humans. The problem with these drugs is they are ridiculously expensive at this point. This is that drug that costs $1,000 per pill, and a full course of treatment can easily cost $50,000. So I don't think any beekeeper would consider spending $50,000 to control the viruses in a bee colony. Now, it could be that someday the price of these drugs will come down to a point where direct control of viruses using a drug may become realistic, but I suspect that day is a long way off. Perhaps more promising is this RNAi approach for virus control. And some of you may have heard about Monsanto's effort in this realm. They are calling it BioDirect. This is technology that they got when they purchased Biologics, which was an Israeli company which is developing an RNAi approach for controlling viruses in bees. I should say that Monsanto and Biologics are not the first people to use RNAi to control viruses in bees. In fact, bees themselves have been using RNAi for millions of years to control viruses. And this is probably one of the key ways that bees manage to suppress viral infections on their own if they can have an effective RNAi defense against viral replication and the virus problem. So just a quick summary about how this RNAi works. So going back to a slide I showed previously, here we have the virus genome, this positive sense single-stranded RNA, getting replicated by an RNA polymerase in the cell that it's hijacked to produce this double-stranded RNA. Now, double-stranded RNA doesn't occur in bee cells or in human cells for that matter very frequently unless it's from one of these RNA viruses like hepatitis C or deformed wing virus. And the RNAi system is designed to detect this double-stranded RNA that's a product of viral replication and target that for destruction. So our cells, B cells, we've all got this amazing system to control viral replication. So you have viral replication up here producing double-stranded RNA, and there's a protein in us and in bees that recognizes this double-stranded RNA called dicer. And what that does is it cuts up this double-stranded RNA to produce these siRNAs, and then these little short pieces of RNA actually form a template for these RISC complexes, which then assemble and then are used to target double-stranded RNA back up here that the virus is trying to produce, and it targets those double-stranded RNAs for degradation. And it essentially stops the virus from being able to reproduce its genome and stops the virus from being able to produce more copies of itself inside the cell. And this is the way bees can naturally overcome these viral infections. So what BioDirect and this RNAi approach that Monsanto is using is they would feed or otherwise provide bees with double-stranded RNA outside of the virus and get this whole system working so that if a virus is present, it will be broken down by this RNAi system, even if the abundance of the viral RNA itself from the virus reproducing is not enough to get it going. So kind of priming the pump to break down these viruses if they're present. And getting back into the molecular biology of this, I'm just going to finish up with how we detect viruses in bees and how we know what viruses and virus quasi-species or strains we've got in our bees. And this really gets at the point that the only way to know if we have a viral problem is if we have a way of detecting it. And unfortunately, detecting it is not easy. It really requires quantitative real-time polymerase chain reaction or quantitative or qPCR. And this will allow sensitive detection of viral presence and measurement of viral abundance. And the way this works is you need to collect 50 to 200 bees, and those bees either need to be alive or frozen very quickly. Bees that are dead, you can't isolate these viruses from. Bees that have been put in a regular freezer, you can't isolate viruses from. You need fresh killed bees, very quickly frozen in order to detect viruses. Then you need to grind those bees up in an extraction buffer. You need to extract the RNA. Then you need to reverse transcribe that RNA to DNA so that you can then amplify that DNA using the polymerase chain reaction, PCR, using primers that are specific to that particular virus or that particular viral strain. So here we have the bee copying its gene, or the virus copying its genome inside the bee. We would just have to translate that to DNA And then once it's in DNA, we will replicate large quantities of that DNA using the PCR reaction. And you just have primers, just very briefly, primers that are specific for this specific viral sequence. And those primers will bind to the DNA. You use a DNA polymerase to make a copy of that DNA. And then this is a way to amplify DNA with each cycle of the reaction. So you start off with 1 copy, then you have 2 copies, then you have 4 copies, then you have 8 copies, then you have 16 copies, then you have 32 copies, on and on and on. You do this 40 cycles, and you suddenly have huge quantities of this particular DNA of interest, and you can actually measure the presence of that virus in there at the beginning of the reaction. Enough DNA that you can then visualize it on an agarose gel here, which has been stained with a DNA-sensitive stain, and you can actually see the DNA on this gel. And if that band is present, that tells you that that particular bee had a viral infection. If that band is absent, that tells you that that bee did not have that infection. This seems like a lot to go through, and it is. It's not a simple procedure. It's probably about 4 hours of lab work to detect a virus in a bee, plus all the specialized equipment you need to do the molecular biology. So it's not, I mean, there certainly are probably hobbyists who could do this, but it's not an easy procedure and not something any beekeeper is going to be doing anytime soon, and really just stresses the need that we need more laboratory capacity to determine viral infections in bees if we're going to know what's present, what's absent in, for the average beekeeper who's not going to mess around with the PCR reaction. So, in conclusion, viruses are really underappreciated problem in beekeeping. They're almost certainly responsible for more colony mortality than they're given credit for. But understanding and giving them their due is really stymied by the fact that we have no really easy way to detect viruses short of an overt infection. And there's still a great need to find easier ways to detect viruses and to learn more about their biology so we really know what they're doing in bee colonies and how much of a problem they are. We are certain that Varroa is enabling and vectoring particular strains of deformed wing virus. And that is probably underlying a lot of the problems that we're seeing in bees today, is this virulent strain of deformed wing virus interacting with the Varroa that are ever present. However, controlling Varroa is probably not enough to control the viruses, because we will still have this virulent strain of virus present in our bees, even if we get Varroa down to 0. So this really calls for a holistic approach. We need to get a handle on the evolution of viruses, Varroa, and bees together. And only by appreciating all the players in the system are we going to be able to breed bees that are better able to survive in our current environment.